Dear students, today we are going to see about the important topic in botany, biomolecules. So we are going to see about biomolecules. So what is meant by a biomolecule? Biomolecules are the substance or a molecule which is produced in a living organism. So biomolecule is a molecule which is produced in a living organism. So it is a molecule which is produced in the living organism. So we know that our biosphere is made up of different types of organisms. So it is made up of a rich source of biodiversity. Starting from the microorganisms, plants and animals, we have rich source of biodiversity. When we do an elemental analysis, so what we have to do is, we are going to do an elemental analysis. So what is meant by an elemental analysis? Elemental analysis is the one we are going to analyze what are the elements which is present in the various biodiversity forms, such as the microorganisms or a paste of microorganism or a living tissue or an animal tissue. So if you are going to take any sample and we are going to find out the element which is present in all the living organisms remains the same. So though the constituents may vary, but you will be having the elements such as the carbon, hydrogen, oxygen, all those will be present in every organism. So if you do an elemental organisms in example for in an microbial colony or a microbial space or in an plant or in an animal, right? So we will be able to find out the elements such as carbon, hydrogen, nitrogen, this oxygen, which will be present in all these organisms. So when we compare an element analysis with the living material and a non-living thing, for example, if you are taking a sample of a plant tissue and we are comparing it with the sample which is present in the earth crust. So for example, we are taking a plant sample, right? You will be having a plant tissue. Then we are taking a part or the soil from the earth crust. So we are going to compare these two. You are taking a piece of tissue and we are taking a part or a soil sample from the earth crust and we do an elemental analysis. We can able to find out the material which will be present in both this plant tissue and the earth crust remains the same. You will be having carbon, you will be having hydrogen, you will be having oxygen, nitrogen, sulfur, sodium, calcium, magnesium, silicon, everything will be present. But silicon won't be present in the living tissue, but silicon it will be present in the soil. So when we compare this elemental analysis, we can able to find out the percentage composition. Percentage composition is the sense you will be having how much amount it will be present. The amount will be present more in the plant tissue compared to that of the soil, because you are taking a small area or a small sample, and when we are comparing in the plant tissue, the accumulation or the composition of the materials will be more in the plant tissue or in the animal tissue or any living tissue than that of the soil. So when we compare this, and they have done a analysis and they have given out with the table. So we will just compare the table of the elements which is present in the living and the non-living substance, right? So you will be having a comparison, right? So we will see the comparison of elements present in living and non-living, right? We will be having the living and the non-living substances. So we are comparing. So we will find out. First we have the element. What are the element which is present in it? Right? We will be having the element. Then we will be having the percentage of weight. Right? Percentage of weight of the elements in earth crust and the living organism. Right, we will compare. Right, the element which is present in the earth crust and the, so we will be having this element. What are the elements which is present in the earth crust and in the living organism? So we are going to find out the percentage of the weight or the percentage of the element which is present in the earth crust and the living organism. So we are going to see about the percentage of weight. Right, already we have written about the elements. So we will be having percentage of weight present in earth crust and the living organism. We will see one after the other. So we will be having this hydrogen. So hydrogen we will be having 0.1 percentage which will be present in the earth crust. Whereas in the human body you will be having 0.5 percentage which will be present. Then after hydrogen you will be having the carbon. So carbon you will be having 0.03 percentage. You will be having it in the earth crust. Whereas in the human body you will be having carbon, you will be having more, 18.5 percentage. Then we will be seeing about the oxygen. Oxygen relatively you know that it will be present more in the earth crust. Right? You will be having around 46.5 percentage, 46.5 or 46.6 percentage. But in the human body you will be having more. Right? You will be having about 65 percentage. Right? 65.0 or 65 percentage. Then we will be having the nitrogen. Nitrogen you will be having, it will be very little. It will be present in the earth crust, and because you will be knowing that the source of this atmospheric nitrogen fixation, you will be having lightning and you will be having the microorganisms, symbiotic forms only, they will be fixing the nitrogen to the soil. But whereas you will be having more amount in the living organisms, you will be having about 3.3 percentage which will be present in the living organisms. And then you will be having the sodium. Right? Sodium relatively you will be having more in the earth crust than in the organisms. Right? You will be having 2.8 percentage which will be present in the earth crust, and you will be having only 0.2 percentage which will be present in the living organisms. Right? So you will be having this hydrogen, carbon, oxygen, nitrogen, sodium. Right? You will be having this percentage. Then we have to see about the calcium, magnesium, silica and sulfur. Right? You will be having this sulfur. So sulfur when we compare, you will be having, which will be more in human body. Right? You will be having 0.03 percentage in the earth crust, and you will be having about 0.3 percentage which will be present in the living organisms. So then we compare about the other things. Right? Since there is no space, I will write you on the top. Right? Right. Then you will be having the substances, right? Calcium, magnesium and silica, right? Si. Right? You will be having calcium, magnesium and silica. Silicon. Right? So you will be having calcium. Calcium you will be having 3.6 percentage. But in the living organism you will be having only 1.5 percentage. Because this calcium, magnesium and silica, which will be present more in the earth crust than in the human body. Right? You will be having 2.1 percentage, you will be having 0.1 percentage, and silica, right? Relatively you know that silica, it is present in the sand, right? And in the you will be having negligible quantity, right? You will be having NeGle, GBle, right? We will be having a negligible quantity of the silica which will be present. So you will be having, when we compare the elemental analysis of the organisms or the elements which is present in the earth crust than that in present in the living organisms, relatively some of the components except this calcium, magnesium and silica, you will be having this carbon, hydrogen, oxygen, nitrogen, this sulfur and sodium. Sodium also you will be having present in the earth crust will be, you will be having more. You will be having the elements are present more in the living organisms than in the earth crust, because the living organisms they might have uptaken in the form of food, or due to this growth process they have accumulated the more amount of the substances than that present in the earth crust, right. So now we are going to see about the percentage composition of elements, right. How to analyze the percentage composition. So in this lesson we are going to see about the analysis of the chemical compositions of elements present. Then we are going to see about the structure of these primary and the secondary metabolites. Then we are going to see about the biomacromolecules. Then we are going to see about the structure of protein, structure of these amino acids, then structure of these polymers, these carbohydrate polymers, then protein trafficking mechanism and the metabolites and the dynamic state of metabolites, the steady state and the enzymes. So we are going to see about the mechanism or the working of the biomolecules in our system. So now we are going to see the analysis. Composition of elements or the compounds. We will be mainly dealing about the compounds, analysis of percentage composition of compounds in living system, right. We are going to see about the analysis, right. We will be having the analysis of the composition of compounds, mainly the chemical compounds, right. We are going to see about the chemical compounds which is present in the living tissue, right. For this you have to take a piece of tissue, right, living tissue, right. It may either plant, right. In case of plant you may take a vegetable, for example you may take a carrot or a green leaf or any tissue you can take, and in case of animal you can take a liver tissue or a epithelial tissue you can take out, right. You may take any type of tissue. So first we have to grind, right. We have to make it in the form of a slurry, right. Slurry is a mixture or it is a form of a semi-solid paste, right. We have to make it as a slurry. So how we will make it as a slurry is, we will take a mortar and pestle, right, mortar and pestle. So mortar and pestle you might have seen, right, in all types of these Ayurvedic forms and advertisements you will be having the one, right. You will be having a bowl like thing, right, and inside that you will be having a pestle which is used to grind, right. You will be having a pestle, right. This is called as the mortar and this is called as the pestle, right. So what you have to do is, you have to take this plant tissue or the animal tissue and put it into this mortar and pestle and you have to grind it. So for grinding you will need a liquid substance for grinding, because for any type of the compounds which has to be analyzed, you have to make, since you have to make it into a paste, you have to mix it with a liquid. For this you are using a solvent, right. You are using carbon tetrachloride, right. C right? CCl3, CCCl3COH, right? You will be having this carbon tetrachloride, right? So this carbon tetrachloride, you are mixing it and you will making it as a paste or the slurry. So in this slurry, once you have made, you will be having a mixture or a semi-solid part, right? You will be having, right? Right, you will be having this carbon tetrachloride, right? So when we made this as a slurry with this type of this carbon tetrachloride, after the mixing it is a paste, you have to filter it, right? You have to filter either with a cheesecloth or a cotton cloth, right? You have to filter. Cheesecloth or with a piece of cotton cloth, right? You will be having this filtrate, right? So you will be having the filtrate, right? You will be covering it, right? You will be having this for it. You will be covering it, right? So with the funnel you are having, you are making it as a filter, right? For example, you will be having, take a beaker, right? You will be having a funnel, right? The funnel, you take a piece of cloth, right, or a filter paper, then you have to pour it, right? This pour the suspension, you will be having the filtrate, right? The one which is in the bottom is called as the filtrate. More technically or more chemically, it is called as the acid soluble pool. And you will be having the retentant or the acid insoluble pool, right? Mostly this acid soluble pool, they will be having the organic compounds, and most of the inorganic compounds are the macromolecules which will be present on the acid insoluble salt, right? You will be having the retentate or the retentant, right? You will be having the acid insoluble pool, right? This carbon tetrachloride, CCl3COH, you will be having this carbon tetrachloride. When you filter it, you will be having the two types of the fraction. One is the filtrate which will be in the liquid form, and another one is the solid part which is called as the acid insoluble form, insoluble pool, right? Right, when you have this filtrate or the soluble part, you will be having the most of the organic compounds, right? You will be having carbon, hydrogen, nitrogen, oxygen, right? You will be having all these will be present in the soluble part, and in the insoluble you will be having the inorganic components, right, or the macromolecules which will be present, right? For this, what are the molecules and the percentage composition you have to do? You have to do an analysis, right? What is this? It is called as the chemical analysis. For the chemical analysis, you will be having, you will be studying it in your higher classes. You will be having the various type of the chromatography, and the centrifugation technique. Right, you will be having the chromatography technique, or you will be having the centrifugation technique, right? In the chromatography technique, you will be having various methods. In centrifugation, you will be having the settling of molecules according to their mass and gravity. You will be having a rotor that will spin very fastly, and based on the spinning, you might have also seen about the centrifugation in your clinical labs. You might have seen small for lab testing of blood samples. You will be having, right? Your blood sample will be there. It will be made to spin. Right? Chromatography, you will be having various type of chromatography. Right? Chromatography, you will be having the column chromatography. You will be having the paper chromatography. Then you will be having the thin layer chromatography, gas chromatography. Then you will also have the HPLC, right? High pressure liquid chromatography. So in column chromatography, you will be having a thing which is called like the burette. You will be having, it will be packed with the materials like silica and alumina. Then you will be having the solvent which is to be fractioned or analyzed. It will be poured, and you will be having the substances like alumina or the silica that will be absorbed, and the other will be read out. So you can able to find out once on combining these molecules, you will be pouring different types of solvents, and different types of solvent have the different solubilizing nature, and it will dissolve the substance, and you can able to see the different fractions, and you can able to find out what type of amino acids or what type of the chemical compound which is present. Right, in paper chromatography, you will be having a mobile phase and a stationary phase. Right, you will be having a thin layer of paper. Right, then you will be having a mark the spots or the sample which is present in it. You will be having beaker which is containing the liquid. This will travel. You will be having the mobile phase. As the solvent travels, it will have the different color or the marking, and based on the color and the shape of the compound, we will be having a chart, and we can able to analyze what type of compound is present in it. Then you will be having the thin layer chromatography. Thin layer chromatography, it's one you will be having a thin layer of glass like that of the paper. You will be having, you have to pack this thin layer of glass, right, either with alumina or silica or any type of substance. Then it will be kept on the solvent, then it will move according to the movement. You can able to find out by spraying different types of chemicals. You can able to find out the amino acids or the components, sugar components which is present in the substance. Then you will be having the gas chromatography. In the gas chromatography, you have to pass the substance in the column which is containing the gas. Right, you will be having an apparatus like this. You will be having letting in. Right, you will be having a column. Right, that of the gas you will be having. When it enters into the column which is containing the gas, you will be having, you can analyze the substance. Then you will be having the HPLC, high pressurized liquid chromatography, which is the most efficient method. Even if you have a sample of about 2 microliter, right, you will be having very small amount of 2 microliter, right, you will be having a solution which is of very lesser in quantity, right, you will be having 2 microliter also. You can able to find out the compound which is present in the, right, microliter, you know, right, 1 ml divided by 1000, you will be having the microliter, 1 microliter, right. So you will be having, even if you have 2 microliter, you can able to find out the component which is present in the substance, right. These are the techniques which will be adapted, right. So you can able to find out the substances, right. In case of the compounds which is to be analyzed for the inorganic components, right. So if you want to find out the inorganic components which are present in the tissue, for the composition of inorganic tissue, we have to burn the tissue, right. For example, if you want to find out the what are the types of inorganic components which is present in a plant tissue, right. For example, if you have a leaf, right, you have to find out the inorganic components. What are the inorganic components present in the leaf? So first we have to take the leaf, right. So the leaf, we will be having it as a wet leaf, or the wet weight you will be having. So you have to make it to dry, right. Once we dry, the water molecules which is present in it, right, it will be evaporated, right, water molecules will be evaporated. After evaporating, we have to burn this tissue. Once we burn all the carbon and the water vapor, right? Along with that, if you have some water vapor also, it will be taken, right? You will be having the carbon and the water vapor will be released, and you will be having the ash. What this ash contains? It contains the sodium, magnesium, potassium and other components you will be having, right? You will be having calcium, magnesium and the potassium. These are the inorganic components which will be present in the living tissue. So if we find out a chart and list out the inorganic components, we will be having the inorganic components such as calcium, magnesium, potassium. Along with that, you will also have some sodium, which also will be present in the living tissue. So if we make a list or a chart of the inorganic compounds, what are the inorganic compounds which is present in? We will see it as a form of a tabular column, right? Inorganic constituents. What are the inorganic constituents of the living tissue? Then you will be having the inorganic constituents of the living tissue, right? So you will be having what are the inorganic components and their symbols, right? Sodium, Na plus. Then you will be having the magnesium, Mg plus plus. Magnesium you will be having it in the ions. Magnesium you will be having. Then potassium, K plus. You will be having potassium. Then you will be having the water. Water molecule H2O, right? Then you will be having the calcium, Ca plus plus. It is the calcium. Along with that, you will also having the some of the compounds, right? Inorganic compounds. It will be present in the as a form of compounds, right? Compounds you will be having NaCl, you will be having calcium carbonate, you will be having. Then you will be having the sulfate, right? You will be having SO4 2 minus sulfates. You will be having. Then you will be having the phosphates, PO4 3 minus, right? These are the compounds you will be having. You will be having the sodium, magnesium, potassium, calcium, water and various type of the compounds, right? Inorganic compounds such as NaCl, sodium chloride, right? Cl2 won't be there, Cl, right? Then you will be having calcium carbonates, carbonate forms, sulfates and the phosphates. These are the inorganic constituents of the living tissue. So more chemically, or if you have a person who is well versed in chemistry, he will analyze the compounds in the form of their chemical nature such as aldehydes, ketones, alcohols or then aldehydes, right? So you will be having a chemist, if you have a chemist, right, who is well versed in chemistry, right? So you will be having, they will be classifying. How he will classify? Based on this classification method, he will classify as aldehydes, right? CHO, aldehydes, all acids, right? COH, or alcohols, right? Alcohols or OHs, right? Acids, right? Aldehydes, or in the form of ketones or any other aromatic esters, right? you will be having the classification which is based on the chemist. A person who is well versed in chemistry, he will classify these compounds as either it is an aldehyde, acid, or a ketone, or an ester, or an ether, or any type of compounds, or a aromatic compound, right? But in case of biologist or a biologist classification, right, biologist classification is somewhat different. It may be classified as a protein. If it is a protein, you will be having amino acid. Then carbohydrate. If you are having a carbohydrate, it is classified into sugar, right? Then you will be having the lipids. You will be having the fatty acids and glycerols you will be having, right? So this is what a biologist person classify the compounds of the biomolecules in the form of proteins, carbohydrates, or lipids. You will be classifying it as an amino acid, or a carbohydrate, or a lipid. So the mainly we are going to see about the amino acid, right? One after the other we will be seeing, right? First we will see about the amino acids, right? You know that these proteins are the building blocks of every organisms, right? You will be having proteins. For every organism, it serves as the building mechanism. These proteins are mainly made up of amino acid. You will be having the amino acid. So this amino acid structure, it mainly contains a carbon. So as you know that you will be having the carbon as a valency of 4, right? So you will be having a carbon which is having the 4 valency. One, it will be substituted by an amino group. Another will be having a acid part, carboxyl group, and you will be having a hydrogen, and another will be having the R group. This R group decides the type of amino acid which will be present in the system. This carbon is called as the alpha carbon, because you will be having the carbon which has both the amino and the carboxylic group. So for an amino acid, you will be having an amino group and then carboxylic group, and you will be having an hydrogen which will be present in the carbon to have this attached to the 4 valencies, right? You will be having the R group, right? Based on this nature, the amino acids are mainly classified into 20 types. You will be having 20 types of amino acids are present. You will be having serine, you will be having valine, glycine, alanine, phenylalanine, tryptophan, tyrosine, and you will be having this tryptophan, phenylalanine, that isoleucine. You will be having various type of amino acids, 20 types of amino acids. They join together to form the proteins, which is called as the building blocks of an living organism. So you will be having, based on these R group, if you substitute hydrogen, you will be having glycine. If you substitute methyl group, you will be having the alanine, and if you substitute the hydroxymethane, you will be having a serine, right? You will see, right? One after the other, you will see. So you will be having this carbon, you will be having an amino group, then you will be having a carboxylic group, then you will be having an hydrogen atom, then you will be having the R group, right? If this R group is replaced by hydrogen, you will be having glycine, right? You will be having glycine, right? If R group is replaced by hydrogen, you will be having glycine. If it is replaced by a methyl group, you will be having alanine, right? For example, you will be having this. If it is a methyl group, you will be having alanine, right? If it is replaced by a hydroxymethyl, group, right? If it is replaced by a hydroxyl methyl group, you will be having the carbon, you will be having an hydrogen, right? You will be having an amino group, you will be having a carboxylic group, you will be having a methyl group, hydroxy methyl, you will be having the serine or the serine amino acid, right? Based upon this, you will be having different types of amino acids, and you will be also having the amino acids which are of acidic in nature, basic in nature, and you will be having also the neutral amino acids, and also you are having the aromatic amino acids, right? These are the one which will be classified based on the position of the R group, right? The substituted R group, you will be having different types of amino acids, and you will also have the amino acids which are of acidic in nature, acidic amino acids, basic amino acids, and you will also have a neutral amino acid, right? We will see about these three. Acidic, you will be having glutamic acid, as in the name itself you will be having the acid, glutamic acid. Glutamic acid is an acidic amino acid. Then you will be having basic, lysine. Then you will be having neutral, valine, right? You will be having the glutamic acid which is the acidic in nature, basic one which is the lysine, and the neutral one which is called as the valine. In addition, you also have the aromatic amino acids. Aromatic amino acids you will be having tyrosine, phenylalanine, and tryptophan, right? Aromatic amino acids. You will be having the aromatic amino acids. You will be having phenylalanine, tyrosine, and you will be having the tryptophan, right? You will be having three types of these aromatic amino acids. You will be having the tyrosine, phenylalanine, and the tryptophan. These are the aromatic amino acids, right? And you will also have these amino acids, they form the zwitterionic form, right? Zwitterionic form is the one you will be having this, right? You will be having the form which is having, you will be having the central carbon atom, then you will be having the CO minus, then you will be having the NH3 plus, right? Then you will be having this R group, and you will also have a carbon, right? You will be having this carbon which will be present in this R group, you will be having, and right now, okay, you will be having this. This is a zwitterionic form, right? This zwitterionic form, you will be having the reconvertible nature. You will be having this amino acid. You will be having this NH3 plus. You will be having it in the form. You will be having where this R group, acidic group, you will be having replaced, right? You will be having this amino group, and the zwitterion is the one you will be having, which acts as a buffer, right? It will be acidic and basic, both will be present. Acidic and basic both will be present in the same group. So you will be having, they don't be, it will be acting as a buffer, right? It won't change in the structure, right? It won't convert into an acid form or an basic form, right? It will be more stable, right? So you will be having this zwitterionic form of the amino acid, right? Then we will see about the carbohydrates or the sugars which will be present, right? You will be having the sugars. Sugars, right? We will be having this sugars. You will be having this glucose and the ribose. Glucose will be having C6H12O6, right? So we have to see about the structure of the glucose, right? You will be having the O group which will be present. Here you will be having a hydroxy group, methane, hydroxy, methane. These two, then you will be having the OH, OH. Here also you will be having the OH, and here also you will be having a OH group, right? This forms the structure of the glucose. Then you will be having the ribose. Ribose, you will be having the structure C5H10O5, right? These are, you will be having a pentagons like structure, right? You will be having, right? You will be having the O group. Then you will be having the structure, right? You will be having a structure, right? 5 ringed structure. Here you will be having CH2OH. Here you will be having, here you will be having a OH group, here OH group, and here also you will be having a OH group, right? This forms the structure of the ribose, right? Like this you will be having the sugars. Examples are glucose and ribose. Like this you will be having the fatty acids. When fatty acids are break down, you will be having the fatty acids plus glycerol you will be getting. So in the next class we will be seeing about the various types of this palmitic acid, glycerol, and you will be having the structure of the adenine, guanine, and the cytosine and thymine structures. These forms the structure of the nucleic acid. Those structures we will see. Then we will see about the protein primary, secondary, tertiary forms of the proteins we will see, and we also see about the polymeric structure of the carbohydrates. Those things we will see in the next class. So today what we have seen is that we have seen about what is a biomolecule and what are the elements which is present in the living system and in an earth crust, and we have also seen about the comparison of elements, and we have seen about the tabular column. You will be having the components are present more in the living system than in the earth crust, and we have also seen about the elemental analysis, how to analyze the chemical composition, right, of a living tissue. The chemical composition, you will be having the soluble part and the insoluble part, right? Acid soluble pool and the acid insoluble pool. You will be having the filtrate and you will be having the retentant, and you will be having the organic and the inorganic components. What are the organic components, right? Organic compounds are the one you will be having the carbon as the predominant form, and those which does not have carbon you will be having the inorganic components. So inorganic components you have to make it in the form of ash. Then you will be having the calcium, magnesium, potassium, and various salts you will be having. Then in the form of tabular column we have also seen what are the inorganic components which will be present. Then we have seen about the mechanism of classification by the biologist and the chemist, right? So what the chemist will do, he will classify as aldehydes, ketones, or the aldehydes, or the acidic, or the alcohols, or the ketones, or the esters. Then the biologist will classify it as the proteins, amino acids. Proteins will be classified in the amino acids, then carbohydrates and the fatty acids, fats, right? So we have seen some of the structural examples of the amino acids and the sugars. The rest of the things we will see in the next class. Okay. Thank you, students.